Hello friends, welcome back to the course Current Regulatory Requirement for Conducting Clinical Trial in India for Investigational New Drug and New Drug version 2. This is our lecture 14 and this is a special concern lecture, and really this lecture covers all the special concern which require special attention as it involves different committees, and other than the regular our product, that is a new chemical entity, it requires certain special attention to be given. So the learning objective from this lecture: upon completion of this session, the trainee will learn in brief about the biologicals, why they are special, then phytopharmaceuticals, stem cell and stem cell derived product, radiopharmaceuticals, regenerative medicines, nanopharmaceuticals, and the different committees involved for the approval of this product. So let us begin with the biologicals. Are they special? Certainly these are the special biologicals. The biologicals, it covers vaccines, biotech derived product, hormones, enzymes and other. Let us see why they are special in nature. If we see for the vaccine, they are derived from living organism, and that could be ranging from normal or genetically modified organism to fluids or tissue derived from various animals or human sources. They frequently have a complex molecular structure. That's why they are special. Then the biotech derived product, as we know, they are having highly diverse molecular structure, highly diverse mechanism of action and the highly diverse clinical indications. The other product like recombinant hormones, insulin, growth hormone, cytokines, erythropoietin, interferon, enzymes and the monoclonal antibodies, these all are the biological products. And the special thing in this, that for the regulatory approval it required to take a new drug permission every time. So as per the New Drug and Clinical Trial Rule 2019, these are the special product, and there may be a change in the quality, safety and efficacy of this product whenever the new manufacturing process is there or whenever there is a new manufacturer, and that's why these products are always considered as a new drug product. Unlike our routine drug, what we have seen, once the drug has been approved, that drug remain new for only 4 years, and after that the applicant need not have to take the new drug permissions, and they have to take the permission from the state licensing authority to manufacture it. In case of this biological product, these are always a new drug product and required to take a new drug permission every time. So let us begin with one by one. First we will see the vaccine. So as I have mentioned, vaccines or these biological products are different from other drugs as they are derived from the living organisms, and they frequently have a complex molecular structure. That's why vaccines are also considered as new drug every time. And the demonstration of safety and efficacy for use in human is essential before it can be approved for import or manufacturing, and this permission is given by the CDSU, that is National Regulatory Authority. The regulation under the Drug and Cosmetic Act and New Drug and Clinical Trial Rule 2019 describes all the information necessary for seeking approval of vaccines along with an application to import or manufacture for market and sale. Let us see what is the biosimilar. The biosimilar is the similar biological or the similar product, and it is a biological medical product referring to an existing registered product and submitted for medicinal product registration by an independent applicant after the time of protection of the data has expired for the original product. A biosimilar product would have an abbreviated non-clinical and clinical development, leveraging on the existing information of the original product and focusing on demonstration of similarity with the original product. Different terminologies has been used for this similar biological or biological, like in EU it is a biosimilar product, in US it is a follow on protein product, in Canada it is subsequent entry biologicals, and likewise. The basic principle involved in this biosimilar product, that a new biological product claiming to be similar to a reference medicinal product, standard generic approach, that is demonstration of bioequivalence, is scientifically not appropriate, and the biosimilar product approach will have to be based on the comparability, that is demonstration of the similarity between the existing product and the new product, more likely to be applied to highly purified product which can be thoroughly characterized. Products such as vaccines, blood or plasma derived product, gene or cell therapy product, these are not acceptable. Let us see what is recombinant DNA vaccine. So, the recombinant vaccine may be broadly categorized into three groups, that is subunit recombinant vaccine, attenuated recombinant vaccine and vector recombinant vaccine. The subunit recombinant vaccine, these are the components of the pathogenic organism. Subunit vaccines include like a protein, peptides and DNA. Attenuated recombinant vaccine are the genetically modified pathogenic organism. Pathogenic organism, we know that bacteria or viruses that are made non-pathogenic and used as a vaccine. The vector recombinant vaccine, these are the genetically modified viral vectors that can be used as a vaccine against certain pathogens. Some of the development made in the production of recombinant vaccine against certain diseases, and the guideline to be followed for the rDNA derived product are these. These are the guidelines what have been shown on the slides, so I am not going to read it all. So, various applicable guidelines have been given, like recombinant DNA safety guideline from DBT, guideline for generating preclinical and clinical data, the guideline of 99, again it is from the DBT. Then CDSCO has also published these guidelines in 2008. There is one handbook which was for the institutional biosafety committee, and again there are revised guidelines which are now in existence from the CDSCO. Let us see what is the difference in the giving the approval to this product. So, we have seen in our chemical entity that the committee involved are subject expert committee, TAG committee and apex committee. After the thorough deliberation with this committee, the product approval gets. Apart from these committees, the other special committees are there to review the applications pertains to this biological and the rDNA. These committees are review committee for genetic manipulation, we call it as RCGM. RCGM is under DBT and it is responsible for authorizing import export for R&D, that is research and development purpose, and it is responsible to review the data up to preclinical evaluation. So, this committee is responsible up to preclinical evaluation. Once the preclinical evaluation is been over, then they give the recommendation to the CDSCO, and according to the for the clinical trial conduct, the CDSCO reviews the application. The another committee involved is a Genetic Engineering Appraisal Committee, that is a GEAC. It functions under the Ministry of Environment and Forest for review and approval of activities involving large scale use genetically engineered organism, that is LMO we call it, living modified organism, and products thereof in R&D, industrial production and environmental release. CDSCO, as we know, it is a Central Drug Standard Control Organization and it is a national regulatory authority under the Ministry of Health and Family Welfare, and it is responsible for grant of import, export, the clinical trial approval and permission for manufacture and market. The various state licensing authorities, they are there for the grant of manufacture and sale license. Let us see the other part, that is a phytopharmaceutical. So the phytopharmaceutical has been defined in the rule 2 subclause AA we have seen in our earlier lecture. These are a purified and standardized fraction with defined minimum 4 bioactive or phytochemical compound. They are qualitatively and quantitatively assessed of an extract of medicinal plant or its part for internal or external use of human beings or animals for diagnosis, treatment, mitigation or prevention of any disease or disorder, but it does not include administration by parenteral route. The data to be submitted along with the application to conduct clinical trial or manufacture of phytopharmaceutical drug in the country, it has been given in our New Drug and Clinical Trial Rule 2019 in Schedule 2, Table 4. We have already covered in our previous lecture. Let's have just look at this data. So this has been divided into the two parts, Part A, Part B. Part A is data to be submitted by an applicant and the human or clinical pharmacological information, and Part B is data generated by the applicant like identification, authentication, source of plant used for extraction and fraction, the process of extraction and other data. Let us have a look at the stem cell derived product. So these stem cells are undifferentiated biological cells with a capacity for self-renewal, proliferation and differentiation into many different types of functional cells. Based on the potency of the stem cell, there are 5 types of cells. These can be divided into the 5 types. The multi-component cells like mesenchyma cell, then hematopoietic cells, then pluripotent stem cell, for example embryonic stem cells or induced pluripotent stem cell. These are pluripotent stem cell. Then totipotent stem cells like a stem cell derived from morula, and unipotent stem cell, which are for example hepatoblast or the cholangiocytes. These are the unipotent stem cells. And the last is the oligopotent stem cells, for example lymphoid or the myeloid stem cells. The classical definition of stem cell requires that it should possess 2 properties, the self-renewal and the potency. Self-renewal is the ability to go through numerous cycles of cell division while maintaining the undifferentiated state, and the potency, it is the capacity to differentiate into a specialized cell types. The ability of stem cell to self-renew and give rise to subsequent generations with variable degree of differentiation capacities offer significant potential for generation of tissues that can potentially replace diseased and damaged areas in the body with minimal risk of rejection and side effects. Stem cell and stem cell based product we call SCCPs are regulated as a drug under Drug and Cosmetic Act section 3B. However, the research on the stem cells and stem cell products comes under the ICMR, and researchers shall follow National Guideline for Stem Cell Research 2017. There is always confusion where to apply, whether it comes under the purview of regulation or not. So by this slide we try to give the clarity. The term stem cells based product is used to refer to product intended to be administered to patient and that contain or are derived from stem cells. At present, the therapeutic treatment by stem cells as provided in the NGSCR, that is National Guideline for Stem Cell Guideline 2017, are considered as established therapies, and rest, as I have mentioned, come under the development of research category. CDSCO, being a national regulatory authority, regulates stem cell based products which are in the form of a product and are marketed for use as a drug, that falls under the purview of D&C Act 1940. And for development, manufacturing, this, as it is coming under the purview of D&C Act, a permission from licensing authority is required. Stem cell and stem cell based product, as I have mentioned, regulated as a biological product under New Drug and Clinical Trial Rule 2019, and as per the GSR 334 dated 4/4/2018, stem cell and stem cell based product means a drug which has been derived from processed cells, including cell or tissue which has been processed by means of substantial or more than minimal manipulation with the objective of propagation and or differentiation of a cell or tissue, cell activation and production of a cell line, which include pharmaceutical or chemical or enzymatic treatment, altering a biological characteristic, combining with a non-cellular component, manipulation by genetic engineering, including gene editing and gene modification. What is meant by minimal manipulation and other things that are also given in this GSR. A separate task force on stem cell research and regenerative medicine has been constituted to consider the proposal on their scientific merit by the Department of Biotechnology. So, apart from for the evaluation of it, apart from the subject expert committee, then technical advisory committee and apex committee at the CDSCO, one more committee, that is CBBTDEC, that is Cellular Biology Based Therapeutic Drug Evaluation Committee. It is constituted by the Government of India, Ministry of Health and Family Welfare, under the chairmanship of Secretary, Department of Health Research and DG, Director General ICMR, to advise the DCGI in matter pertaining to regulatory pathway leading to the approval of clinical trial and marketing authorization for the therapeutic product. Ministry of Health and Family Welfare, wide order number DCGI miscellaneous 2010/2010 part stem cells TFQC, dated 1st September 2010, has constituted a core IND panel of experts, namely Cellular Biology Based Therapeutic Drug Evaluation Committee, under the chairman of the secretary. Let us move towards our next slide, which is stem cell research. So, as I have mentioned, stem cell research is as per the guideline developed by DBT and the ICMR. So, as per this guideline, that is the National Guideline for Stem Cell Research, no stem cell administration to human is permissible outside the purview of clinical trial. According to the revised National Guideline for Stem Cell Research, jointly prepared by Department of Biotechnology and the ICMR, and this has been announced on 11th October 2017. But any stem cell used in patients other than that for treating approved blood, that is hematopoietic like a disorder, is investigational at present and can be conducted only in the form of clinical trial after obtaining regulatory approval. It means it requires a clinical trial permission from the CDSCO. Genome modification, including gene editing of stem cells, germline stem cells or gamete and human embryos, is restricted only to in vitro studies, not in vivo. Only spare embryos or gametes can be used, and genome modified human embryos should not be cultured beyond 40 days of fertilization. According to these guidelines, only umbilical blood bank is permitted for the present, but not banking of all other biological materials until further notification. All international collaboration in stem cell research and import of any type of stem cell require approval from the respective funding agency and regulatory approval from the Central Drug Standard Control Organization. The guidelines prohibit research related to human germline gene therapy, reproductive cloning, and clinical trials involving xenogenic cells, those derived from different species. Breeding of animal in which any type of human cells have been introduced is prohibited. Let us see what is the regenerative medicines. This is also another area of concern. So, the regenerative medicine is a branch of translational research in tissue engineering and molecular biology which deals with the process of replacing, engineering or regenerating human cells, tissues or organ to restore or establish normal function. All regenerative medicine strategies depend upon harnessing, stimulating or guiding endogenous developmental or repair process. Accordingly, stem cell research plays a central role in regenerative medicines, which also spare the discipline of tissue engineering, developmental cell biology, cellular therapies, gene therapies, biomaterial including scaffold and matrices, chemical biology and nanotechnology. When a human body gets injured or get into a disease state including infection, cold or fever, the human body has got the innate immunity to heal and defend itself. However, the innate immune response is slow and not always too effective if it is a very serious injury or chronic diseases. Here comes the regenerative medicine. It has got the potential to improve the healing process of the body on its own and or accelerate the healing process through clinical intervention. Regenerative medicines works toward repairing damaged tissue and organs and to find a way to cure previously untreatable injuries and diseases. So, there are major areas under regenerative medicines. Actually, it is a very broad field as it focuses on the regeneration of cells, tissue or organs to restore impaired function. The field of regenerative medicines can also be categorized in the following 4 subdomains, that is medical devices and artificial organs, tissue engineering and biomaterials, cellular therapies, clinical translational. The 3 major areas are regeneration, replacement and the regeneration, that is 3 R's. The regeneration branch focuses on boosting the body's innate ability to repair itself. Research in this segment aims to improve the self-healing process on a cellular level. Regeneration is one of the most talked about areas of regenerative medicine. This branch focuses on cell-based therapies to help regenerate healthy cells or cell tissues so that optimum health can be archived. DCGI in April 2017 has approved one of the product that is called Chondron. It is a cartilage repair procedure and uses the body's own cartilage cell that are cultured, multiplied and implanted into the patient's damaged joint, leading to a new cartilage generation and avoiding the need for early joint replacement. The another area of concern is nanopharmaceuticals. Let us see what is the nanopharmaceuticals. It is defined as a pharmaceutical preparation containing nanomaterials intended for internal or external application on human for the purpose of therapeutics, diagnostics and health benefit. The nanomaterial is generally defined as material having particle size in the range of 1 to 100 nanometer in at least one dimension. However, if a material exhibits physical, chemical or biological phenomena or activity which are attributed to its dimension beyond nanoscale range up to 1000 nanometer, the material should also be considered as a nanomaterial. Therefore, any pharmaceutical containing such a material should also be considered as a nanopharmaceutical. The guidelines for evaluation of nanopharmaceutical in India has been published by DBT in October 2009. The categorization of nanopharmaceuticals. These nanopharmaceuticals can be categorized depending on the nature and functions of the nanomaterial as well as the approval status of the nanomaterial and the conventional API from the drug. Accordingly, this has been classified into the based on the basis of here the table has been given. The basis for categorization is degradability of nanoparticles fabricating material, and in this the categories are biodegradable, non-biodegradable. The examples are protein, lipid, biopolymers, and for the non-biodegradable relatively less used in pharmaceutical product, they are like gold, silver, platinum. Based on nature of nanoparticles, the organic nanoparticles, inorganic nanoparticles, multi-component nanoparticles, these have been divided. Then basis for categorization, one more basis for categorization is nanoform of the ingredients. The nanocarriers loaded with API, example micelles polymer conjugate, polymer nanoparticle, carbon based material. Then API converted to the nanoform drug may be converted into nanoparticle or nanocrystal, thereby increasing their potential for improved resolution and bioavailability. Example are nanocrystals of sirolimus, tacrolimus and the phenofibrate. Specific consideration for evaluation of nanopharmaceutical in the context of second schedule of the New Drug and Clinical Trial Rule 2019. General requirement as specified in the second schedule of the New Drug and Clinical Trial Rule 2019 will be applicable for all the nanopharmaceuticals. However, due to the inherent complexity in nanotechnology based product, a case by case approach should be adopted for evaluating their quality, safety and efficacy. In case of liposomal formulation, specific US FDA guideline for liposomal drug product of April 2018 may be consulted. Let us see in brief about stability testing of nanopharmaceutical. According to the general requirements specified in clause 5 of the second schedule of NDCT rule 2019 and also in the ICH guidelines, when the drug is loaded in the nanocarrier, the stability of the drug in its active form should be confirmed from time to time under defined storage and transit conditions. In addition, parameters specific to nanomaterial based system need to be quantified at different time interval for size and size distribution, surface characterization, drug loading, drug release kinetics and other using appropriate techniques. In case of surface coating, for example with PEG, the PEG layer thickness or quantity should be measured by appropriate analytical method. The morphology of the nanoproduct should be determined by microscopy. The residual drug in the system with reference to initial drug loading and drug encapsulation, that should be assessed. The next area of the concern is the radiopharmaceutical. So let us see what are these radiopharmaceuticals. Radiopharmaceuticals are unique medicinal formulation containing radioisotopes which are used in major clinical areas for diagnosis and for therapy. As we know that it is a common treatment for the cancer. This radiopharmaceutical can be divided into the four categories, that is radiopharmaceutical preparation, radionuclide generator, the radiopharmaceutical precursor and the kit for the radiopharmaceutical preparations. What are these? That has been given in this slide. You can see this radiopharmaceutical, the last category, that is kit. When they are derived from the kits, are normally intended for use within 12 hours of the preparation. Let us see why these are special. The regulatory permission need to be taken. In case of the radiopharmaceutical also, the regulatory permission is required. As like we have seen, the different committees are involved in the evaluation of the different kind of product and the biologicals. For the radiopharmaceutical, the no objection certificate is required from the Bhabha Atomic Research Center, and as per the guideline, the radiation exposure must be within permissible limit. Pregnancy need to be ruled out. Then radiation guideline must be followed if death occurs with radiation implant, and appropriate training is necessary in handling these radioactive substances. Now moving towards the next area of concern, that is a traditional system of medicines like Ayush and other. So the Ayush is the Ayurvedic, Siddha and Unani medicines which are regulated by Department of Ayush under Ministry of Health and Family Welfare. This has been, some part of this has also been regulated by the department like CDSU, and the other state licensing authorities also involved. Drug and Cosmetic Act 1940 Chapter 4A, it states the provisions related to the Ayurvedic, Siddha and Unani system. So the license for manufacturing and the sales and the standards required, that has been given into the Chapter 4A of Drug and Cosmetic Act. Drug and Cosmetic Rule 1945, the Part 16 describes the manufacture for sale of Ayurvedic including Siddha or Unani drugs. Then Part 16A is for approval of institution for carrying out test on Ayurvedic, Siddha and Unani drugs and raw material used in their manufacture. Part 17 is for labeling, packing and limit of alcohol in Ayurvedic, Siddha and Unani system. Part 19 is for standards of Ayurvedic, Siddha and Unani drugs. One more area of concern from this traditional medicine is homeopathy, and Part 6A of Drug and Cosmetic Rule deals with the sale of homeopathic drug. Part 7A is related to the manufacture, for sale or for distribution of homeopathic medicines, and Part 9A is related to the labeling and packing of homeopathic medicines. So these are the areas of concerns. Let us see what we have seen in this lecture in brief. So in this lecture we have seen what are the biologicals, which are the area of concern, and why these biologicals are special in nature, what are the rDNA derived product, what are the phytopharmaceuticals, then stem cell derived product, radiopharmaceutical, regenerative medicine, nanopharmaceutical, then traditional system, homeopathic system, and we have seen the different committees involved in that. So now it's time for you to give the answer. The first question for you: which chapter of D&C Act deals with Ayurvedic, Siddha, Unani drugs? So the chapter has been given in the Drug and Cosmetic Act 1940. So this is a chapter 4A which deals with the Ayurvedic, Siddha and Unani system. Question number 2, RCGM stands for, you have to give the full form of the RCGM. So it is a review committee on gene manipulation. The third question, you have to fill in the blanks. Pharmaceutical preparation containing nanomaterial intended for therapeutic purpose is called, what it is called? So it is a nanopharmaceutical. So these are the areas of special concerns, and always there is ambiguity about these areas, whether to apply for the license or not, which ministry, which department it deals with. Hope now it is very clear from this lecture and you have enjoyed this lecture. We will see you again in our next lecture. Till then, you take care. Bye bye and all the best.